Okay. So I think I'll start off by telling you guys that Current Biology is one of those journals that usually produces pretty solid research, so it's a good place to publish. So generally when you get to Current Biology, you're doing pretty well. But they have some requirements in them that include this weird graphical abstract thing. And what it basically is doing is saying, summarize your entire paper in one figure. And that's a lot, right? That's a lot to do. And so this is a very simplified one. I don't know how you guys feel about it, but we can come back to it after we have discussed the paper and then see if you feel better about it. Because I think it's a lot of information crammed in there. Okay, so, oh, I forgot to open up my little, right. So what is your general feeling about what this tells us in this paper? Zoom discussions, interesting.
I mean, it seems to just characterize the differences between a couple of documented coronaviruses. Kind of characterizes I mean, that kind of says it all, really. It's just a discussion of the molecular differences.
Okay, so how many, did you guys notice how many people were used to build the SARS-CoV-2 genome?
Well, there was 5 like listed on the sequencing part, but I couldn't find in the paper where it actually said like how many samples they had.
So I think, okay, so if you look at the end of the paper, there is a method details section. This is also one of those that kind of just jumps right into the outcome first. So it's a little bit different than what we've been doing. So if you look at the methods, it says they took the data from Liu et al., L-I-U et al.
What page is this on?
I think it's E1, I think is what it says on my printout.
Yeah, it's E1.
It is.
It's weird because it doesn't have a great page number. So it says it takes...
References. Gotcha.
It's not in the references. It's actually in something called the method details section.
Right, but it's like past the...
Yeah, past the most of it, yeah.
That's weird.
Yeah, it is. And you kind of get used to it, but when you write these, you have to write a little bit of your methods in your results. And that's why it makes it, I think it was Lucy who pointed out on Friday that it was actually pretty easy to read because you didn't get hung up on those methods that they used, right? So they used this paper from 2019 to get some of this data, right? And that data is actually mostly from, so I had to look at these papers before I did this. So these are all the pangolin sequences from Liu et al. Right?
Mm-hmm.
All right. How many bats did they use? How many bat sequences do we have in that alignment there? A lot, right? There's quite a few.
There's 15 or something.
So when I usually tell you guys to do one of those summaries, the paper summary things that help you guide through the paper, I ask you guys, where did they get their data from? And so did you get a sense of where most of this comes from? Did they do most of it themselves?
No, they use GenBank. I've read.
Most of it's GenBank, and CBI is the same database as GenBank. So all that stuff is crammed into GenBank. But what they did that's a little bit different is they took all the stuff that had been done before and then reanalyzed it. So they basically redid the work of the original paper. And that's because they wanted to align the sequences differently, and they had to because they had different samples, and so they had to redo it. Okay, so the original paper, the pangolin paper, didn't map the genome to human. So they wanted to, because they wanted to compare human and pangolin and human and bat, they had to reanalyze a bunch of that stuff. Okay. Okay. So we still don't know how many humans we've got in here, although they show about 7 or so in here. All right, so I think, Lucy, was it you who asked about this figure that's up right now? I assume you guys can see the figures.
Yeah, I asked about that one, but specifically B.
Specifically B. What about A? What does A tell you?
Is that just depth?
What does depth mean, Alex?
Like how many overlaps, I guess, that they got on a certain.
Yeah, so
Right.
it's really a measure of the quality of their data. So the lower those numbers are, the less representative copies they have for a given position along the genome. So the best are sort of towards the end, the 20,000 N, and then some in the middle.
So does the depth mean like the higher the depth, the more certain they are that the data that they, like the nucleotides that they have found at that position are correct?
It certainly gives you more confidence in it.
Okay.
There's arguments of, there are arguments about how much depth you really need, but a mean depth of 7 is pretty high. So
Why do you get much higher depth in certain spots?
So it happens partly because of the ease of accessing it and the type of sequence that's in there. If the sequence is really repetitive, you can lose some of it. The way that they did this, if I remember right, is the way that they, when I say did this, I mean way they actually accessed the genome, is they had to take the RNA, so they capture it and turn it into what they call cDNA, which is the complementary DNA to the RNA, right? And then they sequence those libraries, because that, you can sequence DNA better than RNA. So they have that. So some of it may be an artifact of like if the DNA has a lot of C's and G's, the sequencing can really get messed up. Some of it just might be what got captured when they made their libraries.
Okay.
Okay, so there's a whole bunch of different reasons it can happen, but in general, I think the mean depth of 7 is pretty good for now, for us. So that's all they're talking about with when they look at panel A, is just trying to give you a sense of quality. Okay. So I'm going to switch the slide now to just get rid of that, because I just wanted you guys to see it, and let's talk about like the real results here, because this is sort of more important. These are the two sort of bigger ones and bigger deal, I guess. So I think when you look at this, there's probably more information than you realize in these. So when you look, remember we said on Friday that what you're looking at is the percent similarity between any of the things listed in the box and the pangolin. So the higher they are on that figure, the more like the pangolin they are. So at the first position, they're really close, and then you get variation as you move along. So if it were you describing this, which one's most closely related to pangolin? Well, you can tell me either based on the numbers or just based on what you can see.
The red. Right?
Right. And what is that from?
Humans.
Humans. All right. So when you look at that sample, it's saying Beta Covey Wuhan, which is one of the Chinese isolates. Okay. And then bats are really close as well. So what about the original SARS virus?
Pretty different.
There are a lot of areas, yeah, I would agree. There are a lot of areas that are pretty different, especially in that middle, right above where it says query, sort of in this area here, you get these dips, right? And then around position, I don't know, let's just say 22,000, 23,000, you get a massive dip between bat, SARS, and all the other things that are in there, right? Okay, so what this tells you overall, how would you summarize what this tells you in a general sentence?
The pangolin virus seems to be the most closely related to the virus found in humans.
Okay, so mostly, right? And there are a few areas that might differ, right? So you've got at least here, there's some dips, right? But it's pretty similar. Okay, so, and then when we look at the bottom half, why does this particular thing matter? Why would you show this? Remember I told you it costs a lot of money to print things in color. So what does it matter here? This is very colorful.
So there's like
Well, it
different.
shows the similarity of the specific proteins that are made.
Okay, so that's in those white boxes, right? Like this is your similarity. What else is, but what about this part in the white?
What they don't have
They're
that.
actually
huh?
comparing.
Yeah.
Okay, let's start one person at a time here. I don't care who, just raise your hand or something and then go. All right, Madison, you start.
Isn't it where they don't have the data for it, or they're missing the genes?
They're definitely missing nucleotide sequences there. So when you compare this, the SARS-CoV-2 from human and the pangolin, and you talk about position 0, or let's say 5,000, right? So 5,000 might hit, well, let's say 7,000 because it hits a white area. Does that correspond to the 7,000 that's up in the top graph?
Yeah, mostly. They're
Can
a
it?
little bit offset, but
So if there's no data between pangolin at 7,000 and the genome position 7,000 in humans, is that comparison there?
No.
No, you can't compare it if you don't have sequences for that same area.
Mhm.
So when you get these big dips, right, we're missing some information here because it's really hard to make a comparison to pangolin in humans if there's no data. Yeah, Alex.
oh, so that's what all the like flat areas on the top graph mean.
It's really
Where it's just like
unclear
a-
what the flat parts mean, because those flat parts should be corresponding to an area where there's no information.
Mm-hmm.
So it's a little bit unclear. Right? So when they have this report of 91% similar, that means that of the amino acids and DNA sequences that they have, 91% of those are similar. But if you were to just take 29,000 and 19,000, you're talking about 10,000 base pairs that they don't have information for. Okay, so there's some comparisons that are missing. Okay, does that make sense to everybody? What probably should have happened in this diagram is there should have been gaps where the data was missing. Okay, so that probably is how they would have done it, should have done it. But in any case, they're still showing you there's a ton of similarity as you go along. All right. Okay. So let me ask you guys, well, let's just go ahead and move on to the next part. So I think this stuff actually is a good description of what is in the genome of the pangolin CoV2, right? Like that's not very interesting in terms of evolution anyway, right? So all it's saying is they're similar in some places. But what does this tell you? First of all, what data are they using here?
On the left, they're using the whole genome.
Left is the whole genome, and on the right?
Like a specific protein?
Yeah, which specific protein is that?
Oh, is it the RNA-dependent RNA polymerase?
Yeah, good. So it's always, when you take a look at a phylogeny and you see that they present two, there's usually a reason to present two. So why might it be, why would you expect the whole genome to look like the same phylogeny you get when you look at the RNA-dependent RNA polymerase?
Can you repeat that? I'm sorry, that didn't
Why
really
might you expect the phylogenies to look the same for the whole genome and for RNA-dependent RNA polymerase? In terms of evolution, right? Because this is not just about the disease. This is about like what we can use the phylogeny for.
Because it's a key component of the life cycle, and so it's probably going to be highly conserved.
Okay, so one of the expectations you have of the RNA-dependent RNA polymerase is that even if it accumulates mutations, you'd expect the changes to be neutral, because if it changed too much, it could screw up its ability to be an RNA polymerase. And so you would expect that at least the RNA-dependent RNA polymerase should reflect the evolutionary history better, because it's not going to be undergoing massive changes in its evolutionary history. Because if it does, it may not work anymore. That makes sense?
Mm-hmm.
Okay. So what does it tell us here? What does it tell us is more closely related to humans, pangolin or bats?
Pangolins.
Yeah, absolutely. So wait, which is more closely related to humans?
Wait.
That's
Yeah.
Oh, wait. I didn't see the green one. Sorry.
That's okay. No, it's worth actually kind of taking some time to look at this, because this is, they're saying that the pangolin is the origin of the human virus. And then if that's the case, why would you then see that the bat is more closely related to humans?
The
Right?
bat or the pangolin?
That's what I didn't understand.
Okay, so when we look at these two things, so they've highlighted a couple cool things in here. One, they're showing the bat viruses that gave rise to human SARS, right? The original SARS. They're also showing you MERS down here is in here as well. That's the human coronavirus that causes MERS. And then the original human SARS is just labeled SARS-CoV-SZ3 up here at the top, right? So I think that when you look at this, what you see is that all of the human coronaviruses, the COVID-19 viruses, their most closely related virus is the bat, right? So that's what that means, right? They have a common ancestor at, let me annotate here. So the bat and human share a common ancestor in that really ugly yellow color I just highlighted. And I'll point an arrow at it because yellow is hard to see. Right? So that's where they share a common ancestor. And then that group, the bat and human group, then shares the common ancestor with pangolin here. You guys all see that?
Mm-hmm.
Okay, so what that's saying is that something about this suggests that the pangolin is the origin for bat and human. You guys, that's what those phylogenies would say, right?
Right.
Okay, so just as a quick note, what do these little 100s mean and 90s and all that scattered about the phylogeny?
Aren't they bootstrap numbers?
Yeah, what does bootstrap mean?
Hold on. It's like the number of times you repeat it, you get the same exact layout of your phylogeny.
Right, and so what they're actually doing, so you guys, well, let me just jump ahead because I can show you this really quickly. If we look, oops, that's still annotating. All right. So when we look at data, we look at it in something like this. This is like an alignment. So you can see that just looking at the first position or the position 340, because it's numbered, you see that there's a P there for beta Wuhan human 1, right? And then if you go all the way down, everybody has a P there, right? That's what the dot means. Okay. What they actually do with bootstrapping is they go in and they randomly pick a subset of these columns of data. So maybe 340, maybe 370, 380, and then they rebuild the phylogeny. And they look to see how many times they get the same relationships. All right, so bootstrapping is a way that we look at how good does the data support this, how well does this data support the phylogeny? Or can we randomly remove a bunch of sites and still have the same phylogeny? That's basically what they're doing. Okay, so when you have 100% bootstrap support, that means the phylogeny is well supported with a lot of different data points. If it's not, then that means you may or may not have very good support for the phylogeny. Okay? Okay, so right now, what we see from this phylogeny is that the pangolin is what we call the sister taxa to the bat and human coronaviruses. But I kept the title up here for a reason, right? So they say probable pangolin origin. And that suggests to me that what they're saying is that the human virus came from pangolins, right? That's what that kind of says. But it may not actually be all that they're trying to say there. So go ahead. And Benji, were you going to talk? No.
No.
My phone is making, or my computer is making it sound like people are starting to talk. Okay, so what the heck is this thing? It's an alignment of all the samples, but what is it?
It's comparing the amino acid sequence of the S1 protein specifically.
Okay, why do we care about the S1 protein?
Because it has the receptor binding domain to get into host cells.
Okay, so if you're a virus and you have a specific protein that allows you to enter cells, is this going to be under selection?
Yes.
Yes, because it determines what hosts you can get into and what hosts you can't.
Right. So we have this receptor binding area. So this particular protein. Do you expect the relationships among the samples to look the same as they did for RNA-dependent RNA polymerase?
No.
No.
It might not, right? Your expectation going in is that this could look really different because it's under a different selection pressure, right? So,
Right.
okay, so when we look at this, one of the things they've done that's a little bit confusing, I think, is they've highlighted all of the places where there's a different amino acid. So in order to look at that, if you start with just the first one that has letters in the column, you can see that the humans have an L at that position, and many of the bats do, and even the original SARS do, and the pangolin does. But then some of the bats have a V, and then other bats have an R. You guys see that? Okay, so that's what we're looking at. And so what they wanted to do is take that information and build a phylogeny with it. All right? So what I wish they had done, and I did with my own pens, is to, oops, sorry. I'm trying to get the annotate button to show up for me again. Okay, so if we, so pink is sort of the color for the human viruses, right? So if I were to circle those, they would be here, right? Everybody's good with that? And then the red is the pangolin, and then green is going to be our bat. And so that one's down here. So this is the pangolin, pang, and then this is human. Is this the same as the phylogeny we were just looking at?
No.
No. It's saying something like, well, the pangolin has the same protein at this, or a more closely related spike protein to the human than the bat covey. Yeah, Benji.
The one you circled in green, isn't that the bat?
Yes. Oh, yes, it is. Oh my God.
Yeah. That's how I was a little bit.
No, totally fine. I even said it out loud, right? Like I even said.
Right.
So doesn't mean you speak well when you try to. Why can't? How do I turn the eraser off? Bad. That's better, right? Okay. Thanks, Benji. So this tells us this is a different relationship than we see with the whole genome phylogeny or even the RNA-dependent RNA polymerase. What does that mean to you?
Is there a possibility that the spike proteins like evolved the same thing independently, or probably not?
It's a really good question.
Because I feel like that seems unlikely, but.
So, well, I think it's the right question because we're looking right now at the protein sequence, right? And the protein sequence is not the DNA sequence. So this is really a phenotype, right? So is it possible to get the same phenotype with a different genotype?
Yes.
Absolutely. So this would be an example of convergent evolution if what Isabel is saying is correct. So if they have the same phenotype, they converged on the same phenotype, but have different DNA sequences, then we have convergent evolution. Okay? So the next logical thing to do would be then to go look at the DNA sequences for the spike protein. Right? That would make sense to do, to ask that very same question. So, so go ahead.
Can it be convergent evolution if they have a common ancestor?
Yes, it still can, because if you think about, it just depends on how far back it is.
Gotcha.
Right? So it really depends on how far back it is. I think that's where you have to be a little careful in wondering what the right answer is. But I think it's important to think about the fact that there are two ways to get the same protein, right? There's exactly the same DNA sequences and exactly the same protein sequences, and then there's also different protein sequences to get the same thing, right? So here we've got, different DNA sequences to get the same proteins. Okay, so if you look at the, I don't think I stuck these in the PowerPoint, so hold on just one second. If you look at the supplemental information at the end of this article, I have it in the PDF, so I've got to share that with you, but Zoom, where is Zoom? I love watching me try to find windows on my computer. Let me go to share screen and then pick a different screen, this one. All right, so if we look at, I think it's figure S3, if you have it, they do the same thing. Can you see the PDF?
Yes.
Yes.
Okay.
Mhm.
So in figure S3, you have the same idea. So this is the amino acid sequences. You can see all the same coloring of the taxa anyway. And what you see is that for, wait. Sorry, I want you on figure S2. Sorry. So if you do the phylogeny of the S2 proteins, so on the A on the left is S. Why is this so screwed up? So sometimes you guys will find that the supplemental material is not as well annotated as the stuff in the paper. And what you can see is that it says phylogenetic relationship of the ORF1A and 1B on the bottom here in the figure legend, but then at the top they have them labeled S and M. And then in figure 3, it's supposed to be S and M, and it's ORF1A and ORF1B. So that's a little bit messed up. So I'm going to assume, hold on, I'm looking at something here. This is so weird. So they're arguing that, this is so messed up, you guys. Well, we can't even look at this, can we?
I guess they didn't double check that part.
No, they didn't, but it also is confusing because what they show you is that no matter what, in all 4 of these phylogenies, if you look at the relationship of pangolin relative to the human,
They're all the same.
they're all the same. Now the one on that's labeled figure S1 on the right, it shows that pangolin, bat, and basically those really short branches mean that none of these are very different from one another. And they're also not very different from another bat, one of the blue bats that causes the original SARS.
But they also have those low numbers about their like.
Right, and those low numbers just basically mean we can't tell. We can tell that these guys are they are in a clade together because they have 91% support right under that red dot. But the branches are short, so we can't tell how each of these are related to one another. Right? So we can't tell if bat, pangolin, or human, or how the sister taxa relationships are. So in this case, if it's correct that every single one of these phylogenies, regardless of whether they are the open reading frame 1, open reading frame 2, the S protein, or the N protein, what does this tell us about the way that this spike protein potentially evolved? Is it
Those convergent, because if all of the rest of the phylogenies for all of the other proteins show that the human's more closely related to the bat, then the relationship of the S protein in pangolins and in humans being similar is likely like not because it had the most recent common ancestor, but just because it evolved that way.
okay? So I think Isabel, it's a really good explanation from the data we're given. So this suggests convergent evolution because the remainder of the genome, with the exception of the spike protein, reflects the relationship as bats are the sister to human coronavirus, and then pangolin is sister to that. Does that make sense to everybody? Okay, so we have some information that supports what they're suggesting. So what do you guys see right now on your screen?
The
No.
PDF, like the bottom of the blankish page.
Okay, so we're still seeing that. So I want to stop that sharing and I want to go back to, oh my God. So apparently if you leave PowerPoint while you're Zooming and you stop sharing it, every time you click anywhere near PowerPoint, it pretends that you're still projecting the screen. It's fun. So, so now you guys should just be able to see each other. Is that correct?
Yes.
Yeah.
So let's go back to this now and share that. Okay. Now you should be able to see PowerPoint, correct?
Yes,
Excellent.
ma'am.
Okay. Okay. So when we look at this last figure, what's the big deal about this last figure? Why do they highlight?
The human one has a 4 amino acid insertion that none of the other ones have.
Correct. So we have this novel insertion. So that should be about 12 nucleotides different, right? It should have, the human should have about 12 nucleotides different within the S protein. Does that make sense? Right? Because they're amino acids and they're 4 amino acids. Okay. So we have what they call a motif. Remind me if any of you guys have taken cell and molecular in here. Do you remember what a motif is? Alex?
It's like a region of folding within a protein.
Yeah, so this particular one is a protein motif that allows one of the enzymes, it's a furin recognition site, which allows splitting of the S protein. And that splitting is what allows the coronavirus to enter our cells. So in order for it to be activated and enter a cell of a mammal, that site right afterwards, the RS, has to be cleaved. And for our cells to recognize it, it needs that little SPRR motif. And the pangolin doesn't have it and the bat doesn't have it. What does that tell you?
That it's a newer thing.
It's either new and it evolved within the human host, right? So remember that viruses can enter into all of our cells at any, or into our bodies, but there has to be some way for it to enter our cells. And so when you think about it in terms of evolution, there's kind of 2 steps. One, there has to be the ability of the virus to enter and replicate into a cell, but then it also has to be able to optimize to its host. So this particular area could be the region of the virus that's optimized to humans, right? So this may have evolved within humans. What's an alternative suggestion to that? Where else could it have come from?
Could it have evolved before and that's what allowed it?
Yeah, absolutely. So in terms of the origin, the animal of origin, it could be that this motif is present in another animal. We just haven't found it yet. Does that make sense? So there's 2 options in evolution here. We could say, one, this is a novel evolutionary event that happened while the virus was adapting to humans, or it could be something that came out of another host and we just don't know yet. That makes sense?
Mm-hmm.
So the reason they identified this, while we are in fact identifying the molecular characteristics of this virus genome, we're also finding some clues as to ways to find the host, the original host. And it may be that this is it, but we also may find something that has that SPRR motif. So I get somebody gonna say something, sorry. No? How are you feeling about this paper? Feelings, thoughts?
I feel like it gave us a lot of good evidence, but I feel like there's not a definite conclusion, which is kind of frustrating, but understandable considering how new it is.
Okay. Others? Do any questions arise as to like why they did the paper the way they did or anything like that? What made them choose pangolins?
Because don't they get a coronavirus that causes like foamy, frothy mouth or whatever? Like it's like a kind of similar to what they're seeing in human patients.
Yeah, so about a year, maybe close to 6 months before, maybe in China, a year kind of close to us, there was in the pet trade some pangolins who were really incredibly ill. And so the vets who found that actually cut out many different types of tissue and tried to identify the virus. And so this particular subset of pangolins actually died from their infection. So that's one of the reasons they targeted it. It was making them very sick and killing them. And it doesn't seem to, even the bat version doesn't seem to have any issues with illness from the coronaviruses, even the ones that are really closely related. So hang on, I discovered that there's actually like 10 or so different papers that have come out simultaneously about this exact set of data. And then I just gotta find the right one. So right after we started reading this paper, another paper came out in the journal Nature. So like, what's today? The 30th? Is that right? It's 30th. So last week, the 26th of March, so last Thursday, in Nature, another paper came out, and it did a much better job covering the genome. So they actually found that they used the same tissues, but recreated the genome. And hold on, let me see. I think I downloaded this on like 7 different computers, so just bear with me for 1 second here. I certainly am having some issues. So go new share. I want to go this. Stop share. New share. Now do you see it? Do you see the paper?
Yes.
Ma'am.
All right, cool. So when you go through and look at it, they got about 28,000, I think, bases. So they have almost complete coverage. And if you, let's see how this works for you guys. Does this zoom in for you? Do you get?
Yes.
Excellent. Maybe I should present in this all the time. I feel like I should sometimes just use this. So if we look at their data set, hide the sidebar, go away. Okay, so here's their phylogeny. And so with 30,000, with the full-on genome, this is what their phylogeny looks like. Right. I don't know what you guys are seeing. So is it zoomed into the phylogeny with the SARS-CoV-2 related lineage? So
Yes, ma'am.
So now you see that they have 1, 2, 7, 8 Manus javanica, and that is your pangolin. You've got a bunch of human sequences, and you can see they've labeled that clearly SARS-CoV-2. And then what's coming to be the like standard of all comparison is this particular bat, RATG13. So 13 referring to 2013. RA is the genus species name. And and then I don't remember what TG is, but it's an abbreviation for bat coronavirus that still appears to be sister to SARS-CoV-2. But look at the two sets of pangolin sequences. What does that say about the pangolin sequences? Thoughts, feelings?
Does the length of the bars indicate that they're like less related than the other paper seemed to claim?
It's hard to know because there's a different amount of data there, right? They're comparing what, 20,000 bases to 30. Yeah, so you have 30,000 base comparisons. So it's hard to know if the length really means anything here.
Okay, gotcha.
But just looking at the relationships of your pangolins, what does that say about the diversity of viruses in the pangolin?
It's a lot.
There's difference, right? So you've got this clade of pangolin viruses down at the bottom where they're almost all identical to one another, right? There's no branch lengths there. And then you have these two at the top that clearly belong very closely related to human or bat, right? So what do you think? Why am I showing you this? Because I think, you know, it's interesting that you get about 10 papers doing exactly the same thing, but they're showing you different stuff, right? They're showing you different things. So first, what does it tell you by using more than one pangolin sequence? Anybody? Thoughts?
It just shows that there's, you know, more than one. It's not a uniform virus, possibly.
Okay,
Like within
so
the population, there's diversity.
right. So I think the first thing that we learned back in the first week of this class is that there's variation in populations. And so when you think about the pangolin variation, there's going to be a lot of variation in the types of coronaviruses that they have. So that's the first part, okay? The second part is that there are multiple viruses interacting with one another within the cell, right? So you've got a lot within a single place. So they did effectively the same kind of thing that the paper you read did, but I thought it was cruel to give you the new one the day of. Yeah, Madison.
When you said there's multiple viruses in a cell, is that like comparable to how influenza viruses are with the H1 and?
Yes, absolutely. So influenza viruses, we name our influenza viruses based on their surface proteins, right? And so we have different regions of proteins that we can name. So H1, N1 refers to the surface proteins. And you can have different surface proteins. So in reality, what we could actually be having in pangolin is all these different viruses living together. And so what the other thing that this paper does is it uses, it tries to look at whether or not there's possibility of what we call recombination in viruses. So Benji, when you first asked about the questions you wanted to learn about in viruses, you asked, how does it make that jump to different hosts? That was your question, right? So what they're proposing and what all of these papers are kind of coming to is that when you look at the S protein in pangolin and you compare that to the human S protein, what they're starting to think is that there was a recombination event either between bat and pangolin or whatever this thing is that entered us and pangolin viruses.
Like a co-infection and
Yeah, so co-infection refers to having multiple different strains of viruses or multiple different parasites at the same time. Okay, so if that happens in mammal populations, what you end up seeing is this potential for different parts of the virus to exchange with one another.
right. It's like similar to crossing over, kind of, right?
It's almost identical to crossing over, and we can talk about this on Wednesday because we're out of time today. But what I wanted to kind of lead you into is the fact that because there's this conflict in the phylogenies, we actually may be seeing some evidence that these have recombined. And so on Wednesday, what we'll talk about is how that happens in viruses. We know the most about flu, so we have to use flu and the types of variation in flu to do it, but we can talk about this, okay? All right, is that cool? And Christy, I promise on Wednesday we'll answer your question about the drugs that are potentially HIV drugs being used for this treatment of COVID-2. Because I did look it up, but I wanted to make sure we had time to talk about this first. Okay? All right, cool. And so if you guys want, I can upload this if you want to take a look at this particular paper, and I will see you guys on Wednesday. All right, Benji, go to class. I know you have another class.
Thank you.
Bye all.
Bye. Does anybody have any questions left, or you just want to go?
Thank
Good.
We're good.
What'd you say, Isabel?
Thank you.
All right, bye.
Thank you.
I just said thank you. Bye.